Dr. Siddiqui has been associated with CSIR as a scientist since 2004 and is currently working as chief scientist. He did his graduation in human biology from AIMS New Delhi and his master's and PhD in biophysics from AIMS. He carried out his postdoctoral research in the field of computational biology and chemistry at University of Würzburg, Germany, and National Research Council Biotechnology Research Institute, Montreal, Canada. His research interests are bioinformatics, chemoinformatics, and artificial intelligence, machine learning driven rational drug design. Dr. Siddiqui has notable awards and honors to his credit, including BMBF Fellowship in 2000-2003 by Government of Germany, NSERC Fellowship by Government of Canada in 2003-2004, the Scientific and Technological Research Council of Turkey Visiting Fellowship in 2018 and very recently in 2023. He has delivered more than 75 invited talks in national and international conferences and workshops, including prestigious ICTP UNESCO conferences on drug design and development. He has been part of the conference held at Italy ICS UNIDO on drug development for developing countries. Dr. Siddiqui has been a constant visitor to CSIR South Africa. He has been a visiting consultant at ICS UNIDO Trieste, Italy. He is a visiting scientist at Ankara University, Turkey, where he has made visits in 2018 and 2023. Dr. Siddiqui himself has been organizing a number of training programs and workshops in the area of bioinformatics and computer-aided drug design and has supervised about 20 PhD students for their doctoral research. Dr. Siddiqui has, to his credit, more than 200 research papers in reputed international journals, and Dr. Siddiqui has been a continuous source of encouragement for bioinformaticians in this part of the world, that is the northern India. He is one of the key workers in the field of bioinformatics. We welcome you, sir, and we would like you to start with your talk, which would be very much informative and encouraging and maybe an eye opener to the beginners.
Thank you so much, Dr. Nidhi. It is really it's a wonderful opportunity to be here again in a program that is organized by your institute, and of course this is highly useful, it's a very important topic that you have selected for this year's FTP. I must thank you for being part of this event. I can see my friend Raghu also in the audience. I must thank for supporting these type of events. He has been a good acquaintance for many years now, and I know his contribution to this development of this field in India. Good to see you, Raghu. So, the topic of this FDP, I think this is very apt, and also it is very timely that we discuss how things are moving in the area of computational drug discovery, how it is transforming the classical computational chemistry to AI, and then now we are discussing things on quantum computing. So let me begin my formal presentation. I'm not going to be it is not going to be a technically complex presentation. I believe that many of the faculty participants from various organizations are not exactly in this field. So I'm not going to talk complex mathematical equations or something like that. It's going to be a simpler version of to try to familiarize our colleagues with what is happening in this field in more general terms. So let me just try to Okay, can you see something at your end? Dr.
Yes, sir.
Ebi,
Yes, sir. Yes, sir. We are able to see.
Yes, sir.
You have to
I
just
think it's not
maximize.
in the maximize it, yeah. Okay, so is it fully visible now?
Yes, sir.
Okay, sorry about that. So now we know that in recent times, computational procedures are widely adopted in drug discovery and pharmaceutical industry, and most of you are well aware about this fact. Now we know that it's not an something which is it has become an indispensable tool in long process of drug discovery and development. It provides options for understanding chemical systems in different ways, yielding information that is not easy to obtain in laboratory analysis with considerably less cost and effort than experiment. So it is already a well-established scientific discipline, as we know. It is critical in the development of new treatments, has created enormous opportunities to speed up and rationalize the multidisciplinary drug discovery process, and provides us novel approaches to design and development of drugs. Well, the main objective of computational drug discovery is development and application of integrative AI, both machine learning as well as deep learning, also the bio and cheminformatic methods that can be translated to support drug discovery process by designing novel drugs, and with the ultimate aim of improving the human health. So, if we recall, go back into the recent past, not so distant, we used to do modeling using these something like what you see on your screen is these plastic models. I'm sure many of you must have worked with these, some toys with spherical attributes representing different atoms. These are colored by different atoms, and then there are cylindrical rods which represents the bonds between those atoms. And with these, you make some molecules by making combination of various atoms and different combination of different atoms, and by building you make a, well, these were actually the plastic models that we used to do modeling in earlier times. And these physical models were mostly based on molecular connectivity and about the stereochemistry of those molecules that you are working with. They were, they gave some impression of the molecular dimension and also about the shape of the molecules that you were dealing with. However, they were not very accurate. They were difficult to manipulate and also store. So suppose you have a box of those physical attributes and you have to make a model out of that. You can use only limited number of models by using those plastic models. And you make a model, you have to disassemble it and then try to make another model using the same set of atoms and the bonds. And that way, so it is difficult to manipulate also. Suppose you make a complex model and you made a wrong selection of atoms. In place of nitrogen, you put a sulfur or some other atom. So you have to dismantle the whole model again and try to build it again from the beginning. Often fragile and static in the terms. You make a model and suddenly it falls on the floor, and then you have a problem that it breaks, or one atom goes in different direction, other in different direction, and so the model were sort of little fragile and not very robust. They gave very little information about the energy and other properties looking at these models. So the chemical representation was really limited by if you used to build models using this
Sir,
method.
could not see the presentation, sir. Someone is presenting and annotating. Sir, could you please share your presentation once again, sir?
Your presentation is already there.
Ah, yes, sir. Yes, sir. Now it's visible. Thank you, sir.
So these models, they give very little information about the energy and also other properties that are associated with these models. So we see that the chemical representation was very much limited using these models and very difficult to construct large models. So these models were good to build small models like 4, 5, 6, smaller model. But if you want to make a want to build a larger models, then you have a problem by using these physical or mechanical ways of building models. Now today we rely mostly on the computers. Computers allow us to do the molecular modeling. Modeling allows access to the molecular information, and these are useful for predicting properties and as well as how you use these properties for various applications. So whether a molecule has a chair or a boat conformation, you can simply use the chemical programs and try to see what are the properties associated with this. You see how a protein is formed with a different set of chains, how it's what is the tertiary structure of this, like how a protein interacts with the DNA or RNA, or many ways. You can simply use a computer using molecular modeling approaches and try to learn these things in a more simpler way and a simpler and less complex way as compared to the earlier versions when we used to do build models using other mechanical ways. So why do we need to build models in first place? Well, this is true for any field, not specifically for drug design. It is true for any field where the three-dimensional shape of molecules and their properties are important, can usually benefit from modeling. So, for example, the aircraft manufacturer, they simply cannot go, they do not go directly into the workshop and start building their aircrafts. They do lots of modeling involving every dimensions, every aspects that is involved in creating an aircraft and building, bringing it into a reality. For example, what is going to be the dynamical properties, what is going to be the tail spin, what is going to be the elevation, wingspan, everything has to be calculated, predicted, modeled, even before even a single screw is being made, put into place in a workshop. This is true for the car manufacturer also. They plan, design, everything. They do all sort of modeling before actual product is born. And a more simpler example, a township, you go and try to find where is your flat and how does it look like. The town planner, the planner will show you the flat A with the sample ready flat. Will see how is the location, what is the orientation of kitchen, balcony, whatever. And so you give a good real presentation of what is going to happen when that is actually going to be implemented. So scientists use models to see how things work before they can make the final product. And this is safer and less expensive, as we assume that will definitely help you losing lots of money in failure of any project. So modeling actually involves computation and the graphics. Typically, a modeling exercise can have prediction and visualization of shape and the properties of molecules. Molecules could be macromolecules as well as small molecules. You can compare the shape, various properties, how a molecule A is different from molecule B with a simple change in a group. For example, a loss of hydroxyl group in a one molecule can alter the shape and the properties of the whole molecule, and uh, one could be a blockbuster drug and there could be a simple failure. And so you can compare how are these differences in the shape and properties can be evaluated. You can examine, predict the molecular interactions, the reactions between molecule A and a protein, molecule A and with nucleic acids, and so on. You can investigate unstable and excited molecules, modeling of dynamic systems, vibrations, diffusion, and one. So don't worry about these technical jargon. So simply consider that basically modeling aids in understanding the prediction of molecular phenomena underlying any given system. So that was the computation. One of the important aspect of this modeling process is how do you visualize these models, and so that the computer graphics comes to your rescue. This allows you to visualize the models. You can easily alter, display, you can change atom, you can rotate bonds, color properties. So for example, in this panel you can see there is a guy in the red color. Now, suppose this molecule, this residue is an important part which plays a role in binding of a, so this represents a animation of a binding site of a protein. And tyrosine is one of the important aspects. Suppose tyrosine is an important residue in this binding site. And if you mutate this tyrosine to phenylalanine, and that tyrosine is supposed to be important for binding to to a drug or drug-like molecule, the loss of hydroxyl group will make the molecule ineffective. So you can simply click change mutate tyrosine to phenylalanine and see how it is going to affect the binding. So you can easily alter and display it, change atoms, rotate bonds, color properties. And so the spatial representation has tremendously improved as compared to the previously used models, like I mentioned in the physical or the mechanical models. And in a way, they are really highly interactive. You can simply sit in terms of your molecule and you can command that I want to change it to, and I want to change and mutate it to some other residue and try to see effect how it is have on the overall complex. So the chemical properties represented mathematically by evaluating equations in a computer program, AI machine learning models have been achieved, and good chemical representation achieved as compared to the previous ways of modeling. Now we go back slightly back around 10, 12 years back. If you recall, there was a Nobel Prize in Chemistry awarded to these gentlemen, Michael Levitt, Martin Karplus, and Ariel Warshel. And the headlines of the newspaper was that three get Nobel for taking chemistry to the cyberspace. And what does that mean? That means that chemical, if you visualize a chemical experiment, you will visualize a chemist holding test tube in one hand, another a test tube and flask in other hand, and mixing reactions, and you will see certain fumes coming out of those reactions. So this is how we will visualize chemistry. So the chemical experiments ideally take place in labs, but the ability to carry them out in real time through computers, they have won Leavitt, Karplus, and Warshel this Nobel Prize for Chemistry in 2013. So today, the computer is just as important a tool for chemists as the test tube. That was the Royal Swedish Academy of Sciences that said in 2013 while awarding these three gentlemen this prize. Their research in 70s helped us to understand how exhaust fumes are purified or how photosynthesis takes place in tea leaves and so on. This kind of knowledge makes it possible to find the best design, things like new drugs, solar cells, catalytic converter, and many, many other applications. So chemicals earlier used to create models of molecules using plastic balls and sticks, which is now done in computers. And the award of these three gentlemen in 2003 highlighted these aspects and acknowledged why the computers are so important in today's era in this field. Also in 1972, there was another Nobel laureate, American biochemist Christian Anfinsen, and many of you know that in his 1970 Nobel Prize acceptance speech, he said, and I quote, "One day it would be possible," he said, "to predict the three-dimensional structure of any protein merely from its sequence of amino acid building blocks. With hundreds and thousands of proteins in the human body alone, such an advance would have vast applications, offering insights into basic biology and revealing promising new drugs." And so what he prophesied in 1972, this was materialized in July 2021. While attempts to develop computer-aided models capable of solving the protein folding problems have been ongoing for many decades, but the accurate protein prediction has actually eluded researchers until these two seminal papers published nearly simultaneously in Nature and Science in July 2021, which represented the AI-driven software program called AlphaFold 2 and Rosetta. And I'm sure all of you now know what are these. Respectively, they can churn out accurate protein structures by thousands and advance that realizes Anfinsen stream 50 years later. So what this gentleman prophesied in 72, it came into reality thanks to the AI-driven software programs and all those advances that have taken place over the past those many decades. And so after 10 years or 11 years, those three gentlemen got the Nobel Prize in chemistry. Again, in 2024, this Nobel Prize in chemistry again went to these three gentlemen, David Baker, Demis Hassabis, and John Jumper. And this year's prize was about proteins, that is life's ingenious chemical tools. They cracked the code for proteins' amazing structures. David Baker has succeeded with an almost impossible feat of building entirely new kind of proteins. And both Hassabis and Jumper, they developed an AI model to solve a 50-year problem predicting proteins' complex structures. And these discoveries hold enormous potential, as we see and also discuss in later slides as we progress further. So why do we study the protein structures? What is so special about the three-dimensional structures of a protein? Well, the structure of a protein is directly related to the protein's functionality, probably when determining it. And so the reasons for research in 3D structures are, one, the most foremost in medicine, and the reason why we are holding this faculty development program. In the medicine, we can understand how biomolecules function and also interact, and underpins efforts to develop new drugs, in medical treatments, and understand and treat diseases. We can find targets for docking drugs, and these are one of the important aspects. I think you will hear more on these during subsequent lectures from various other experts in this FDP. And also there are other applications in agriculture or industry, which we are actually not particularly bothered about at this stage, and we'll focus mostly on the first application. Now, you must be wondering, how do you get these three-dimensional structures? Well, there are three biophysical methods which helps you in studying or getting the structure of these proteins. One, many of you might be well aware of this X-ray crystallography method. And this is an important biophysical method where you get a three-dimensional structure of a target macromolecules with proteins and nucleic acids, how a ligand binds to a macromolecules, what is the ligand binding orientation, and so on. So you crystallize a protein and diffracts an X-ray, a pattern is obtained, and this is translated in an electron density map, and this is fitted, and then you get an atomic model in the coordinate system, which translates into a three-dimensional model that you visualize in a through a graphical interfaces. Then there is a nuclear magnetic resonance spectroscopy. This is used for determining structure and functional groups of ligand, which are likely to be important for binding. Here you use the solution structures of a protein, nucleic acids, as compared to the crystallized structure which you use in X-ray crystallography. Besides these two, there is a cryo-electron microscopy method also, which is used for solving structures of transmembrane proteins, where it is difficult to get the solution or a crystallized component. So you use the cryo methods to look at the three-dimensional properties and structures of those. Based on these three-dimensional, these three biophysical methods and the data that you generate about the structures, you also do a molecular modeling or homology modeling based, where you predict the theoretical prediction of three-dimensional structure of a protein. So example, so the AlphaFold is one example, or Rosetta, and then of course you have a classical models, a modeler, or various other programs that help you in studying these three-dimensional features of a protein. Of course, AlphaFold is worth mentioning here. This is an AI system which is developed by Google DeepMind and predicts the protein three-dimensional structure from its amino acid sequence. And it provides a high level of accuracy, which is mostly competitive with the experimental predictions. And one interesting fact is that in Cas14, AlphaFold was the top ranked protein structure prediction methods by large margin and produced prediction with very greater accuracy in terms of the prediction with very high accuracy. And of course, there is a Rosetta software which has a very numerous application in de novo protein design or enzyme design, ligand docking, macromolecular complex studies, and so on. So now, if we have been discussing proteins, why? Because this is a part of what we call as a protein structure-based drug design cycle. Now, in this protein-centric structure-based drug design cycle, what you do when you start a drug discovery project, you look at this disease, what you are working on, you look at all the genetics, the physiological, every aspect of disease, and then based on various pathways, the role of proteins, you make a choice of a protein target. Once you make a choice of a protein target, it's about its utility and its importance in any disease process. You try to find about the structure of that protein. You use crystallography or NMR or homology modeling and various, including AI methods. Once you have a protein structure, you can use screening of a compound database against that protein target and try to find if there are certain molecules that fit complementary. They show some complementarily territory to binding to the protein active or binding site. And then in the way you score them on the basis of some scoring function in the binding site, and then rank and try to test them in your experimental setup. On the other way, you look at the binding site of a molecule and try to use some sort of the features that represents the binding site. You try to design something completely de novo without any information about what kind of molecules could be made here. So de novo means that you are building something from the scratch. Here you use the generative AI approaches also to build your molecular scaffolds. You use different fragments, try to link them together and make a composite molecules, and then try to synthesize this if it is synthetically feasible, and then test in the system. Once you reach at this testing stage, you try to see if it is showing some activity. You look, designate them as lead compounds, look at the structure of the protein ligand complex, try to see how these molecules can be further improved in terms to improve affinity, specificity, other pharmacodynamics properties, toxicity. So this goes in sort of iterative way in number of cycles until you find something promising in testing system, and then it further progress into later stages in drug discovery cycle. So how do you get these 3D structures? Well, there is a data bank. Many of you know that it is designated as a protein data bank, and this can be reached through rcsp.org/pdb, or you can also find links through Entrez or Blast or some other links which can lead you to this database. Now, this is a repository of protein three-dimensional structures, more than 200,000 protein structure files from various sources, from various organisms, from humans, from disease-causing, pathogenic, non-pathogenic, every source solved through either crystallography or NMR. They mostly find their way into this protein data bank. So suppose we work on a protein structure at CDRI or NMR structure in CDRI in any pathogenic or human proteins, it finally, the coordinates of those protein generally gets deposited in 3 dimensions in this protein data bank. So the structures from the coordinates from this data bank can be downloaded and then can be visualized manipulated with various imaging programs like KIME, Camera, ASMO, and so on. There are a number of programs that can be used for playing with these molecules nowadays. So if you look at the growth of PDB over a period of years, then see from late 1970s, it has grown enormously. And currently there are more than 200,000 protein structure files. And so that shows that the structural data that is available for us is so important that helps in progressing the overall drives the whole drug discovery process in structure-based manner. Also, it is important to know here that function of many proteins is to bind some target molecules or a set of target molecules and then perform these some functions. And some of you from medicinal chemistry may agree that one of the basic tenets of pharmaceutical chemistry is that the biological activity of a molecule is dependent on the three-dimensional placement of specific functional groups. And we call them as a pharmacophore. So in a molecule, the whole molecule may not be important, but there are certain atoms, groups, or pharmacophoric groups that are important and helps in providing the whole molecule their activity. And by understanding these protein ligand interactions, these are mediated by these pharmacophores. Also, we also know that with the very few exceptions, the drugs interact with macromolecular targets. And these are most often with the specific binding sites of membrane-bound or nuclear receptors, enzymes, transporters, or ion channels. So mostly the drugs have to interact with one of these entities here. Now, if you look at this relationship between the drugs and the macromolecular targets, you can think of this as a relationship between a lock and a key. Here, the lock is your target, the key is your drug. So most of the drugs snugly fit into the target. So once you know the three-dimensional structure of a protein, and if you know the binding site of your protein, that gives you the information about the keyhole where you have to fit your key. So like you fit a key into a keyhole space, you fit a molecule, a inhibitor or a drug into the space which have a specific residues oriented or presented in such a way that it will make a specific interaction with the molecule there. So the like a key that fits into the keyhole, the molecule also snugly fits to the target and then have their action. So this PDB gives access to the structures of some locks, but not all. So if you go to the PDB, you find which lock you are going to work for your protein drug discovery project. And mostly you can look at the PDB search and try to see if the protein of your interest, the structures of that protein are there in that. You can analyze this protein structure, and analysis will provide the details of the lock. By understanding the lock structure, a suitable keys can be designed and identified using the computer programs. And this, in layman terms or common terms, we call as structure-based drug design. So you have a structure, the log structure from X-ray, NMR, or from modeling. You use computer softwares, algorithms, generative AI, and so other methods, and then try to find something which is a best binder to this, and that can act as a drug or as a key to your log by this approach. So this is what we call as a rational drug design, where the three-dimensional molecular structure is one of the important aspects and it forms the foundation of this drug design approach. And this relies on the knowledge of, you can classify them as a receptor-based approach or a ligand-based approach based on the knowledge of the structure of the target protein. And once you have, if you rely on the knowledge of the structure target protein, you can classify them as a receptor-based approach, and you can exploit these receptor then for molecular docking, virtual screening, de novo design, and many other applications. And suppose you have only the ligand information available from you, the activity data about the ligand, you can use these ligand-based approaches for using for modeling 3D QSAR, AI, machine learning-based methods, and so on. So we'll see some of these in more details in later slides. So the idea of structure-based drug design is to identify a better key for a given law. And this you can do by using what we call as virtual screening. Virtual screening means that you are doing something in silico. This involves computational filtering or a docking of a large body of molecules, which is a collection of a database. So you have a various scaffolds, different diverse collection of molecules or binders, and you have a binding site characterized by the various features in the protein. So the idea is to segregate binders from non-binders and then get hands-on to those which have a best binding, fit into the binding site. So if you can see in this example, in one of very earlier PhD project in 2002 by Priyanka, so she used Plasmodium falciparum dihydrorotate dehydrogenase. This is a protein from Plasmodium falciparum. So the active site of the protein you see here is the three-dimensional structure of a protein. The active site is characterized by these residues that you are seeing popping up here. And once you characterize the binding site, you blast a series of molecules into the active site. And so you see here the molecules getting filtered into the active site, and these actually are getting scored by some sort of scoring function, and these scoring function rank these molecules on the basis of their affinity and scores. And so you pick those molecules which are better which are predicted to have a better binding affinity in terms of scoring to the protein, and try to pick those few binders and take them into the experimental assay. So here idea is to identify those binders that have a very high probability of activity in the biological system. So instead of simply assaying thousands of molecules, wasting money and resources, you make a rational effort to select only a few of these which have a higher probability of showing activity in a biological assay. Now, one way of understanding these interaction with the protein and ligand is what many of you are definitely familiar with, this term called molecular docking. Now, this phenomena explores the interaction that occur when a ligand could be defined as any atom, a ion, or a molecule which attaches itself to the docking site of another molecule, which usually is a larger protein, which is known as a receptor. So again, here I am going to bring another analogy, much like the space shuttle that docks with the space station. So you have a small molecule, a Discover or some other space shuttle goes up into the sky. There is an International Space Center, or maybe Shubham Shukla goes up, taking a ride up into the into the space. There is a International Space Center already stationed there. There's a bigger partner. So you wouldn't imagine that this smaller molecule, it cannot simply go and sits anywhere on the space station. There is a designated docking site on the space center where this shuttle will go and binds in a particular orientation, and only then if it fits into that snugly into with the International Space Center, as you see here, there is an interaction between the sets of cosmonauts and astronauts and so on. Similarly, you have a receptor, it is a docking site, and the molecule, to be a good drug, it has to, or any binder has to go and dock at that particular pace in that particular fashion in a highly specific and selective manner. So by understanding this interplay between the ligand and its receptor, we scientists are able to design drugs that would block the docking sites of the receptor and thus disable the functional properties of the protein. And ultimately, this technology would help us to cure some of the today's most devastating diseases. We'll see some examples as we move on. So mostly these docking programs uses the same principles to calculate bound structure. They look for the sites of interaction between ligand and the target. Besides, earlier we classified them on the basis of algorithm that we used to calculate these sites of interaction between ligand and target. So this fast shape mesh matching algorithm used by dog and UDOG, and incremental construction algorithm, FlexX, hammerhead, SLY, and so on, taboo search, genetic algorithm, which is mostly used in gold, Autodog, FlexiDog. Autodog is also one of the very popular tool that is being used by academic communities worldwide. Then you have Monte Carlo simulations, Affinity, MacDog, and so on, distance geometry based docking. And so don't worry about these. These are just to give an idea that there are so many programs use different sort of algorithms, both classical, quantum, AI-based approaches, and so on, and help you in understanding how a protein interacts with the ligand a protein interacts with the ligand, and whether you can study their properties and evaluate them as a drug or drug-like molecules. Now the protein is there for docking molecules. You should have some databases also. Now these databases also have progressed over the past 15 years, more than 15 years, from being a mere repository of the compounds to being a powerful research tool for discovering new leads compounds. And the growth of compound databases in drug discovery, also research fueled the growth in commercial software for chemical database management and publicly available compound repositories. I'm sure Dr. Raghu will give you more detailed flavors of all these things in his presentation tomorrow. Maybe I think I see in the timetable. Well, most of the sources of the molecules that we see here are actually derived from natural sources. So 70% of the drugs that are currently in use owe their origin to the natural sources. And these are from plants, herbs, bacteria, invertebrates, marine organism, and so on. And then you have another pharmacophage ligand libraries. These majority of the pharmaceutical companies have relatively large chemical libraries, which represents the cumulative synthetic efforts of medicinal chemistry within the company. For example, in CDR also, we have our own pharmacophore-based library, which is developed by various medical chemists who have been working in this area for many, many decades and have a collective synthetic database of various molecules. And also a newer aspect of compound libraries is the creation of libraries with maximum diversity, and so you have a diversity-based ligand libraries. To give you some example how you get molecules, for example, from natural plant-based, product-based drug discovery for screening of natural product-based libraries. There are numerous large collections of NPs available for virtual high-throughput screening and development of AI/ML models. One example is Coconut. This is a collection of open natural product. This is an aggregated data sets of the 700,000 elucidated and predicted natural products collected from open sources and web interface to browse. There is another catalog of NPs from Zinc database, AfroDB, Analytic.com, ICC, Indofine, Impact, Nibble, TCM, Taiwan, and so on. It's composed of over 80,000 entries, and which can be directly purchased for your screening projects. And more importantly, there's a diversity screening libraries which can be used for various screening projects, like from ChemDiv, a set of drug-like compounds based on the concept of targeted diversity for hits finding screenings. Then you have Enamine, Real, and Made, Ottawa Chemicals, Thermo Fisher, life chemicals, arsenic, Cambridge, Weybridge, and many, many also. They have millions of compounds that are available in SDF format and can be easily screened through various tools against a designated targets in your project. Now, to give an example of how these methods are then used in real life in labs, for example, I give you an example from how a pharmacophore-based virtual screening can be used, was exploited to identify some human DNA ligase 1 inhibitors in my group a few years back. And this is an important enzyme with implication in cancer. If you know a set of molecules and how they bind to a protein, there is a way you can develop a pharmacophore model based on which important the atoms these are actually play a role in binding to a protein. And so you translate those atoms into some sort of pharmacophore query, represent that this could be a donor, atom, acceptor, and so on, and then define certain, you can restrain those groups by adding some distance constraint and so on. Once you make a pharmacophore model, you screen that against a library, here in this case, a Weybridge library, which is a collection of around 50,000 odd molecules. You screen them and try to find out which of these 50,000 molecules actually fit to these type of features. And once you shortlist them, you screen, look at what is the binding consistency of these with respect to the model, how these molecules are in the shortlisted molecules, how these are placed in relation to the protein, and how they make interaction with the protein. So based on this, you do all those profiling, identify, select few certain top ranking hits for experimental assay, and then you identify some scaffolds which actually inhibits the protein, and then you identify those scaffold as the lead molecules and try to further go into iterative fashion that I mentioned in this protein structure-based cycle. So the activity gives you the confidence that your molecule are actually not a theoretical molecule, and they actually translate into a activity in an experimental way. Similarly, in this CHEK1 protein targeted pharmacophore-based virtual screening, the structure of CHEK1 was downloaded from PDB. FDP-2025 model is generated based on the binding features of a known molecule, screen a database, shortlisted molecules. So starting from 50,000, you come to say only 1,000 probable molecules, and from those 1,000 you further analyze and try to see which of them actually can be taken up into the experimental assay, and from there you pick up those experimental weights and try to optimize them in your further optimization cycle. Another example is how you identify PKNG. This is an serine threonine protein kinase, which is important for a crucial role in regulatory process within the mycobacterial cell and signaling cascade or infected host cell. And so if you know the protein and how it binds to a set of molecules, so we have our own inhibitors designed in the chemistry department. Those molecules, they show some activity against the protein, and those pharmacophore features derived from this were used to identify a new scaffolds which mimics the features of the active molecules. And then you dog them, identify, score them, and then find out which of them are actually going to be taken in the experimental evaluation. And these ultimately lead to you some of the molecules which show good micromolar or some micromolar activity in the assay and are further optimized. Now, so far, this is mostly the classical computational chemistry approaches. Now, with the advancement of AI/ML also methods, with the rapid explosion of genomics in chemical big data, we have seen AI-enabled machine learning and deep learning. They have become indispensable tool for drug designers to mine chemical information and from large compound databases to design drugs with important and precise biological properties. And they have certain advantage as compared to those physical models that rely on explicit physical equations like quantum chemistry or molecular dynamic simulation, machine learning. These approaches, they mostly use the pattern recognition algorithms to discern mathematical relationships between empirical observations of small molecules and then try to extrapolate them to predict chemical, biological, and physical properties of novel compounds with desired features and activities. So how this is integrated into identification of some inhibitors. Now here, for example, K nearest neighbor or naive Bayes random forest SVM, these are various machine learning classification models. So you start with, so for example, here is a HET is histone acetyl transferase. This is an important epigenetic regulator with implication in cancer disease. Now, these are novel therapeutic strategy for treatment of cancer. So with information about known inhibitors, you can design study for identification of new inhibitors using multiple. So modeling models were based using multiple machine learning classification methods. Once these models are done, these are evaluated and best models were used for screening databases. Consensus hits are identified, dogged, and you select best 10 hits based on this for biological evaluation and further take up into and look at how these binds to the protein and how these are evaluated in the experimental assay to further optimize them. This is another example of MTB in oil acyl carrier protein reductase. This is here again, and integrated machine learning models have been used, and these models were then used for screening libraries, identifying molecules. Of course, it goes through a thorough computation analysis, even simulation studies, and before we select molecules for biological assay. And this ultimately leads to those molecules which are further optimized. And some of these molecules which have optimized have actually led to a more advanced stage of development in IND range, where we have predicted scaffolds and being filed as an investigational new drug and has been awarded mission projects to further work on this to improve the efficacy. More recently, the artificial neural network-based modeling for identification of biological evaluation of BASE1, and this is a protein which is beta site amyloid beta precursor protein cleaving enzyme. It's a transmembrane aspartic protease, and this has shown as a potential is a possible therapeutic target for Alzheimer's disease. Now, this is an aggravating disease, involves aberrant production of beta amyloid plaques in BASE1, which catalyzes the rate-limiting step by cleaving the amyloid precursor proteins and generates neurotoxic amyloid beta protein that aggregates to form plaques leading to neurodegeneration. So here, multiple ANN models were developed based on the available inhibitor information about the base inhibitors from multiple repositories, including Campbell, PepChem, and the consensus prediction based on various models were applied, molecules were dogged, identified, and further picked the best molecules that are taken into in vitro evaluation to identify a low nanomolar inhibitors out of these. So this workflow utilizes a multi-stage virtual screening protocol for identifying potential base inhibitors by employing multiple AN-based models. Mostly the hyperparameter, these are tuned models, were assigned a task to virtually screen libraries and use a set of molecules consistently predicted and taken up further. And the best molecules were found to be in the IC50 range of 0.043 to 0.05, really sub-micromolar. So this shows how computational predictive models actually translate the virtual molecules into the real molecules when it in terms of when it comes to the translational aspect of these approaches. Aldose reductase is also another important enzyme of all pathways and is responsible for aggravating diabetic neuropathy complication. Here, more advanced three-dimensional convolutional neural networks-based ALR2 inhibitors. These are classified by techniques using the snapshots of images captured from three-dimensional chemical structures with multiple rotation as input data. So Kushagra worked on developing these methods, and this was convolutional neural network-based architecture was trained on around 360 sets of image data with along each axis, and further prediction on Mayberry's library for each of these models. And subsequently, the biological evaluation and showed up 5 of these molecules as a promising ALR2 inhibitors, and suggested new scaffolds for further optimization in this project. So if you look at the study of the success story of various structure-based drug discovery methods, then you will come across certain molecules like captopril, which was developed by targeting angiotensin converting enzyme for treatment of hypertension developed by squid, or HIV protease targeted lopinavir, eluviran, or nelfinavir viracept. These are antiviral developed by Abbott and Aguran in collaboration with Lilly. Then there is another blockbuster for zanamivir, marketed by name of Relenza. It's an influenza neuraminidase. It is an antiviral developed by Monash University in collaboration with Glaxo. Wellcome. So these involve lots of computational inputs while developing these molecules through these pharmaceutical companies. And more recently, when the AI has started making more inroads into the drug discovery process, we have seen lots of rapid advancements, developments. For example, in early 2020, there is a company called Exscientia. They announced the first ever AI-designed drug molecule as an immuno-oncology treatment for several types of tumors to enter human clinical trials. In February 22, in Silico Medicine, another AI-based company, they reported the start of phase 1 clinical trials for the first ever AI-discovered molecules for idiopathic pulmonary fibrosis based on an AI-discovered novel target, all done at a fraction of time and cost of traditional preclinical programs. Details can be found at insilico.com. Later on in January 2023, AppScience became the first entity to create and validate de novo antibodies in silico using generative AI approaches. In the same year, the FDA granted its first orphan drug designation to a drug discovered and designed using AI to in silico medicine for global phase 2 trial for the drug for idiopathic pulmonary fibrosis. And so you see that the race is already growing. And we have seen a host of AI tools are impacting nearly every stage of drug discovery process, offering huge potential to reshape drug discovery research with the promise of lower cost and shorter development timelines. We understand that AI-enabled drugs discovery offers massive potential to enhance the development of new drugs and to treat potential incurable conditions. So I end up with some of these interesting but harsh facts. You know that the traditional drug discovery is highly laborious and costly process, with average cost to bring a new drug to the market is estimated to be about more than $800 million. Excuse me. Mostly generally, a new drug generally takes up to 10 to 12 or 15 years to progress from discovery to launch, and over 90% of the drugs that entered into clinical trials fail to make it to the market. This is harsh, but a fact, harsh fact, but it's a reality. But over analysis of over past decade or so, it has have shown the impact of computational modeling on drug discovery. We have seen that all these methods speeds up the key steps in drug discovery process by combining aspects of structural bioinformatics, structural biology, structure-based design, and now AI/ML approaches. And so the application of all these modeling in drug discovery and development is expected to reduce the annual cost of developing a new drug by nearly 33%. If you reduce, and also the time taken for drug discovery by nearly 30. So if you reduce the time by 30%, that is from average 10 to 12% to say 6 to 7%, of course, that the money that goes into this also gets cut by almost similar. So the example of like HIV protease inhibitors, they have came into market in nearly 6 to 7 years of research, and that highlights how the overall process of modern drug discovery is being reshaped over the past many years. So I think I should end here. It's almost 1 hour now. I'm happy to take questions from.
Thank you so much, sir, for your talk. Now, the audience, you can ask any of your queries to sir. He would be more than happy to answer those questions. Please, you can ask questions one by one.
I know I kept it a very highly paced presentation, but I think many of you will